bands. So to verify that the band that we were seeing was true, we did the staining in either the Kamasi blue or the silver stain. Now, generally, statistical analysis was performed with ANOVA, and then Fisher's LSD was used for post hoc testing, and the appropriate covariates were used when necessary. Now we're going to start with objective 1, and if you'll remember, that is where we injected adult and juvenile Xenopus laevis with E2 to develop our methodology. For the E2 injections, the animals were kept in individual aerated plastic storage boxes in 3 to 5 liters of dechlorinated tap water. Juveniles were fed 3 to 4 pellets of 1/8 inch frog brittle, and adults 2 pellets of 1/4 inch high protein fish food. Here you can see the boxes that we kept them in, and here's the aeration that is going to them. This is just a representative. This is not all the samples that we actually, or all the individuals that we actually exposed. It's just what we put in the picture. Now, for the injections themselves, the treatments consisted of an E2 treatment and a negative control. The E2 treatment injections were given intraperitonially at 30 grams of E2 per gram frog and a corn oil carrier. And the negative control received only 10 microliters of corn oil per gram frog. One adult female was included as a positive control because we knew we would see patellogenic occur. And then a second injection was given after 72 hours because you get an initial peak that'll stimulate patellogenic production, but then it'll decline. And so a second injection is given to ensure that the patellogenic production stays high enough that we can detect it in the blood. Blood was collected both before and after injections to test to see if it was induced. A positive induction was defined as no band at 170 to 220 kilodaltons using procudamine and before injections, and then a band at 172 to 220 kilodaltons visible in procudamine after injections. Now we're moving on into the results for this. Vitellogenin was detected in the plasma of Xenopus laevis after E2 injection that was not present in before plasma samples. Molecular weight of vitellogenin in the plasma of the Xenopus laevis ranged from 180 to 230 kilodaltons. Here you can see the procudamine gel stain. Here we have the positive control of the adult female. You can see that there's no band here and a band here. Here's our molecular weight standard. And here the blood is listed as before and after. This was the negative control. You can see there's no band there. And here is before and after again, where you see no band and then band and no band and then band showing reduction. This is the same gel in most of blue R250, and once again you can see the same pattern is kept. And you can tell the molecular weight standard where our approximate range for vitellogenin was. Now, here are the adult female tested positive. Vitellogenin was induced in 80% of injected juveniles. There were 2 mortalities during this treatment, neither of which was a result of the treatment itself. One of the frogs actually escaped and desiccated. So, three of the individuals had vitellogenin in before and after samples, and as a result were omitted from statistical analysis. And the reason for this is that because it was in before and after, we cannot count it as either induced or not induced. In females, you can actually have an increase of vitellogenin in response to estrogen chemicals in the environment. So, because we cannot count them in, they were omitted from the statistical analysis. And in conclusion, for this particular experiment, we were able to successfully induce it in E2 injected vitellogenin, and we were able to successfully detect that induction with procudamine. The phosphoprotein band that we did detect was in the range reported in the literature for Xenopus laevis. Now, once we have developed this, we applied this method to the E2 aqueous exposures. So you'll note here that we're moving to the second objective of this experiment. For the E2 aqueous exposures, juveniles of approximately 1 year in age were kept in 10-gallon glass aquarium and 20 liters of solution. They were fed 3 to 4 pellets of 1/8 inch frog griddle, and temperature was maintained at 22.6 degrees Celsius. For the treatments, it was a 2-week exposure with water change being performed after 1 week. The negative control was dechlorinated tap water. We had a solvent control of 0.005% DMSO and an E2 treatment of 1 part per million E2 and 0.005% DMSO. Each treatment had 2 replicates and there were 3 individuals per replicate. We kept approximately the same mass in each tank. We collected blood once again, both before and after exposures, to show reduction. For the statistical analysis, we compared the controls pooled and unpooled, and individuals with phytalogen present in both before and after samples were once again omitted. Phytalogenin, for the results, phytalogenin was induced in the plasma of Xenopus aqueously exposed to E2. Once again, we had 2 mortalities before we could actually collect blood at the very end of the exposure or after. And all but one of the solvent controls had blood in both before and after samples. And so now you can understand why we had the pooled controls. It took away, I had 1 sample for solvent control to compare with, and that is why we had to do that. One of the negative controls actually appeared to be induced, but this could be also because as the juveniles are growing, they reach their own stages for phytalogenin production, and this one may be had become female or started phytalogenin production during that time frame. And here are the percent of the Xenopus laevis for phytalogenin induction in E2 aqueous exposures. The only statistical significant differences that we saw were between the E2 treatment and the solvent control. There were no other statistically significant differences. However, what you can note is that the standard error and the mean here for the negative controls and the solvent controls do not actually overlap 0. So when you add them in, you get 0 induction. However, with the estrogen treatment, you'll note that its standard error is the estrogen treatments will not overlap 0, indicating that this is likely an induction. So for the conclusions for this objective, we did conclude that phytalogenin was induced in Xenopus laevis aqueously exposed to E2, and that although we did not see statistical differences, the mean and standard error indicated that there was actually an effect. Now we are moving on to objective 2, because now we have developed the methodology